Shyam, you can start.
All right. Welcome, everyone, to the computational panel. We have a terrific lineup of panelists here who will introduce themselves to you in just a minute. And we have 40 minutes, so we'll get right to it. As you know, computation is the last mile of the human cell atlas, and it's very complex, and in terms of methodology and pipelines and so on, it's very important that we get this last mile right. So thrilled to introduce or bring up our very diverse panel here, representing many different kinds of expertise. Muz, Vinicius, Christine, Nir, if you could quickly introduce yourselves. Muz, maybe you go first.
Hello, everyone. I'm Haniffa. I'm based in Newcastle University and welcome to Sanger Institute. I'm a clinician scientist, not a computational biologist.
Thank you, Muz. Vinicius?
Hello, I am Vinicius, a biologist, but with a PhD in bioinformatics. I am a group leader at the University of Chile.
Thank you, Vinicius. Christine?
I'm at the University of Melbourne in Melbourne, Australia, and I'm a computational stem cell researcher.
Thank you, Christine. Nir?
Hi everyone, I'm Yosef. I'm from UC Berkeley. I'm doing computational biology, computational genomics with applications in immunology primarily.
Welcome, Nir. Welcome to all four of you. My name is Shyam Prabhakar. I'm from the Genome Institute of Singapore, former physicist and now systems biologist and computational biologist. All right. If you're listening, please put your questions up and they'll be voted on, and we'll address them in the last 10 minutes. But for the first 30 minutes or so, we'll go around the room, go around the panel, pick on some questions that hopefully will be very relevant for this discussion. So to start out, I want to get everyone's thoughts on what has happened last few sessions and yesterday. So basically, which opportunities from day one and earlier today can computational biologists make progress on immediately? And so what are the important unanswered questions in our field that we need to be addressing right now as a community? And Muz, I'll go to you first.
So my own thoughts from watching yesterday was, and also my own experience is this sort of, how to speed up annotation and actually coming to a consensus as a community. And I think it would be very interesting when the community annotation project that Peter Kachenko was talking about yesterday will go live for us to try and test this platform. And I think that would be enormously productive. So that was something that I was very excited about after yesterday.
Thank you, Muz. Thanks for bringing up annotation. And we'll come to that again, I think, later. Vinicius?
Hi, Shyam. Well, there are several aspects that must be addressed, like gene and cell annotations, as mentioned by Musa, the standardization on metadata, how to define the completeness, and another key aspect is the human variation, how to deal with that, how to deal with the reference. Should we have a reference, a unified reference? Should we have multiple references? How should we deal with that?
Excellent questions. Christine?
For me, the big challenge is really starting to extract additional functional descriptions out of the data collections that we have, not just what cells we have, but what makes them that cell. And I think the biggest challenge that we've got is that many of the methods we use to annotate gene sets and understand pathways have been derived at the population level or the tissue level. And we really need to be readdressing that at the level of the single cell. And that's exciting to me because we're starting to understand now, or we have the potential to anyway, how networks actually work at the level of a single cell.
Excellent point. So we have annotation, we have completeness and diversity, we have function. Nir?
Yeah, so I have to mention the annotations as well. I mean, you could see that the discussion moves from speaking primarily about integration to the annotation problem. It's very related, but still it can be posed in a different way. And we'll talk about it, I guess, in a little bit. But just to say one thing here, that I think one big challenge is really to better integrate the automation and the manual effort for annotation, which I think is key. Another set of challenges, maybe to add to the list here, Shyam, is now moving to the big N, to the large number of donors and what can be done with that, which opens again a slew of new things for us to think about.
Excellent point. Yeah, there's very different questions that come up when you have cohorts rather than individuals. And that's a nice framing for the rest of our discussion. And thank you for hitting all the notes, very succinctly. So then the question, a couple of perhaps maybe broad questions. So if we get to standards, okay, because as an atlas, that's part of our role to define some standards that support the field, maybe even define the field. So, and annotation, I think default people think about cell annotation, but you also want to annotate data sets, right? So what are the minimal metadata standards that you all think we need for constructing an atlas, right? You know, we can say age, sex, but is that enough? What other kinds of metadata would we want as minimal standards for our atlas? Anyone who wants to take that?
I can take a first crack at this. I think that in terms of metadata, what's important is the minimal requirements so we can reproduce, that the results can be reproduced entirely if you look at the study, right? So the critical covariates that have been used in the study, either as nuisance or as covariates of interest. So these are key, right? So if you can imagine what are these covariates that I've used in my analysis in either way. So those are the most critical ones, and those are the ones that it's our responsibility, right, to make available in the clearest way possible.
Vinicius?
Yeah, this is worrisome because if you know, there is an estimation that less than 25% of the provided single cell of the studies, less than 25% provided the metadata information containing the cell types, methodological information, and related findings associated with the published data set. And it compromises the reproduction and validation of the findings. I believe that they must have sample level metadata and also cell level metadata, such as sequence library type, cell isolation method, sequence instrument used, the coordinates of the missionality reductions used, cluster assignments, cell type assignments, and other types of attributes that could be included. For example, the cell cycle stage or particular treatments or conditions. And also very important is to motivate data repositories and journals to consider this information in their submission guidelines. And also the reviewers to ask for this kind of information.
Thank you, Vinicius. Chris?
Look, I would love to see people record in detail how they actually isolated the cells, because those methods have such a big impact on the transcriptome or binding of antibodies if you're using hashing. So it's really important actually to understand those parameters, even things like time that has been spent in the isolation protocol would be useful. The other thing I wanted to give a shout out to are these new data frames, things like Annotation in the ScanP project, which let you capture multiple layers of annotations. And this is really useful because it means that you can propagate information from the publication or the original clustering, and you can retain that while adding your own annotations from your own obviously superior clustering and annotation method. And it's this ability to promulgate information between these layers which will let us get to ground truths. Otherwise, we're just going to spin our wheels and keep reinventing approaches to these problems.
No question. Certainly the sample prep will have a huge impact. Muz, you wanted to add something?
Yeah, I was just going to add that, I think it's always important to have as much metadata as you can, but one has to also be slightly pragmatic about how much information is actually, one you can actually get within the constraints of the, hospital setting or whatever the patient resource setting. So it's quite a difficult one to say what is the minimum standards, because the minimum standards in one place will be very different from the minimum standards of another place. And I think the more we have, the better. And if we can have a system whereby we can layer on, the depth of the metadata, that will be a way of kind of pragmatically approaching this problem rather than to just define a blanket minimum standard for everyone.
Yeah, I think it's quite challenging. You're right. First, gathering all the data is challenging, and then compiling it in the right format so it's machine readable is challenging. And then writing some software to parse the metadata people submit and then say this is correct, not correct. And that software can be tripped up by just 1 comma extra or something like that. So certainly, I think a lot of work needs to be done in decision-making and in software implementation. And hopefully, the community will roll out this kind of solutions, software solutions and metadata standards of this form. Along the same or similar lines, okay, we talked about annotation in the opening question and cell annotation, let's say for starters. So you can do it in a sense as this is a definitive annotation for this cell, for each cell in the atlas, there's a definitive annotation. And how would one define that and standardize that? Or say Peter's talk yesterday, we talked about an ensemble of annotations that are, say, crowdsourced. Will that be sufficient? Do we need a combination of both? And it's really the annotation, like Chris, you were saying, everyone will do their own clustering and come up with a new kind of annotation. But an atlas has to be prescriptive to some extent. How do we solve that?
Obviously there's going to be, at some level, some identity to the majority of cells that we capture. But there also needs to be some comfort with ambiguity as well. And I guess it depends on what scale you sit at within that atlas. So for example, you could probably classify a T cell or a myeloid cell with a high degree of certainty, but classifying the state that that cell is in, or perhaps inferring what it's seeing, you might have much more ambiguity around that particular aspect.
I want to raise one point actually about the idea of that there is a gold standard to annotation. And in many ways, in many situations, I think that's probably not quite true. And you could look at it as like a living annotation, because with more information you get in the future, you might actually change how you consider, a piece of knowledge that will change how you actually consider how a cell should be annotated. And then this obsession that the minimal unit should be the cell that we annotate, because, cells probably can be quite malleable and plastic in terms of adopting the type of function that they need given a particular situation, and completely transform and acquire new properties. And whether it's sort of, what is the kind of like denominator, so to speak? Is it actually the cell, or is it actually, thematic gene programs that should be the denominator? These are all very interesting questions. But if you use the thematic gene programs as the denominator, it comes back to Christine's earlier point of how good are currently, functional gene annotations are, because a lot of these terms are somewhat meaningless. I mean, they're quite broad. And as a biologist, I'm often extremely frustrated because it just turns out with very vague terms that I don't even know where to take things further.
It's worse than that, because actually most genes, most protein coding genes in the human genome are not annotated to a pathway. And we rely, therefore, on ontologies which have been inferred from the motifs in the proteins of the open reading frame. We need to do better on that front too.
So Chris and Muz, you've outlined quite nicely how labile annotations can be, how uncertain they can be, how difficult they can be to encode and communicate in any particular ontology that everyone understands and accepts. So those are the difficulties, and it's really, I feel, one of the things that's holding the field back. And as an atlas, there will be people coming in to the atlas just saying, look, just tell me what the cell is, okay? I know it's complicated, I know it's difficult, I know it may change, but tell me something, because presumably you know more than me. And at least some users of the atlas. And maybe I'll toss that to Nir or Vinicius. So how do we start to address that? Like, what do we tell people about the cell? And how do we decide what to tell people about that cell?
No, I think that the pragmatic approach of having an annotation, something we can work with, to just understand the data, what in this or other study, of course, that's necessary. But you mentioned in the beginning kind of going back to Peter's talk yesterday, the vocabulary here, right, and the standardization of the vocabulary, and I think. So after Peter's talk, we also heard about the cell ontology project and so on. And I think that there are some terms there, things that at least at a certain level of resolution can be used to standardize the way that we approach things and define things. And that's quite important because a lot of the conversation now is about these cross-study analysis, cross-network analysis, and so on, things that we can actually put together, transfer knowledge, or really integrate things to get more statistical power and or do some, some additional analysis and so on, right? So I think that that's important, right? Going to the higher resolution, I think we can have more freedom there and do things this way. And I just want to add one more thing to that, is that thinking about this question of, okay, let's say we have this minimal product, but we also have this additional more high level, high resolution annotation. How can we integrate it? So I think there is a place for data-driven approaches, really to think about how we take those manually curated ontologies and actually try to merge them by looking at transitional similarity. So that's I think opens a way for some interesting questions.
So that's a plug for the community annotation. But yeah, a good point also that the coarse annotations can be unambiguous and the finer annotations can be more ambiguous. Anyone else have anything to add to this point?
I just wanted to add one aspect that we should not forget in identifying in annotation, I think, even with the coarse grain, we must make sure that we reach out to the communities that have already worked in this, particularly pathology, because all these words are there in terms of finding shared vocabulary. I'm not entirely sure that we really necessarily have to reinvent everything.
Including pathologists in the conversation. Excellent point. Very important. Okay, let's move a few more questions to get through in the next, say, 12 minutes. This is a topic, I think Vinicius, you brought this up, if I remember, and came up a lot yesterday. So it seems there's a consensus, we need a diverse atlas, and that's a great aspiration. So then computationally, what does that mean? What do we need? What does a diverse atlas look like? An atlas of human diversity that covers the major axes of human diversity. And also, what are those axes that we should prioritize initially?
Yeah, as mentioned yesterday, it will be important, firstly, to avoid something similar to what happened to the polygenic risk scores issues related to the underrepresentation bias of particular populations. So we can target analysis for smarter decisions associated with different diseases. And there are other key aspects to consider as multiple references to avoid the bias of adopting only one main reference. I don't know. This is something that must be discussed. And if so, if you believe that we need to have many references, how many references should we have?
Good points. Essentially, you bring up the point of representation. We need diverse representation in the atlas. Let's say we've solve that problem. And arguably, that's not a computational problem. It's upstream of computation, let's say. Right? So let's say we've solved that problem. We have diverse data sets in the atlas. How do we convert those, computationally convert those diverse data sets into a diverse atlas? Like what would that even look like? What would be the mathematical representation of, let's say we wanted to cover age diversity, for example. We want to say, what do PBMCs look like across all ages, for example? And here's a reference atlas of what PBMCs look like across all ages. What mathematical or data form would that object take? And how would we calculate it? Or is it, I know it's a crazy question. I don't have the answer, but if we want a diverse atlas, we need to get to that point.
It's a really hard problem, Shyam, isn't it? Because we've got to think about not just proportions of cells, but proportions within scalable tissues, if you're thinking about fetal to adult. I don't know. I guess we're going to be in for a really fun ride around new visualization approaches. And I guess animation is going to become incredibly important here, because the static views that we're used to seeing really will only let us look at very shallow slices of something that we hope is very, very broad.
We will need new representations, definitely. Sorry, someone spoke up?
Yeah, no, I just want to add here that with these diversity, right, integration here is the name of the game, and you know how to be, and this is I think going to be our big challenge, exactly how to do it in a way that we are retaining the interesting variation, but still are able to put things on common coordinates so we can give them similar names and make comparisons between them and not just compare like just things that are very separate from each other, right? So here this really talks about how the mathematical object is going to look like, right? So you imagine some, I mean, one way to think about it is imagine some modeling strategies that have this capturing these covariates that are critical, right? But are maybe able to generalize and expand the data or project the data under covariate X or covariate Y and so on. So we can see this, so for example, age. That's a very good example. It's extremely challenging. I don't think we are there and able to do it yet, but hopefully with enough data, this will open up as a possibility.
I might chime in with a kind of like a perspective as a clinician, because this concept of what does a BBMC look like across the age? I mean, you know, you've got the full blood count or the complete blood count, as the Americans call it. And, you know, you have the distribution. And if you go above or below, then you are either, you know, off the standard deviation or below the standard deviation. And I kind of think simply like that in one way, as like distribution curves. I mean, you know, that obviously the data is far more complex and has many more variables and parameters, but you could conceivably, you know, build on a similar type of model and concept.
That's a beautiful analogy, Muz. And I'm remembering the height and weight growth charts that I looked at for my daughter as she was growing up, an analogy of the complete blood count. So yes, I guess for transcriptomes, for example, the equivalent would be that at each age, we define a distribution in gene expression space and say, at this age, this is the distribution of transcriptomes in gene expression space. At that age, the distribution shift to this point and so on. And I think what you're suggesting is similar to what Fabiano was suggesting yesterday as essentially probability distributions in some latent space or some manifold. Quite a complex, one level above of complexity compared to what we've been doing so far, I think. But I can't think of any other obvious way to represent these distributions. So, Chris?
There are going to be some structures which only appear during fetal development and disappear during particular times in childhood, you know, the thymus, for example. And then you go through obvious periods of puberty where you get emergence of new tissues and new structures, and you have structures that cycle, not just through your whole lifespan, but, you know, with a periodicity. How do you model that?
It's very true. It's not monotonic. Absolutely. We have our work cut out for us. Okay, let's move on. 2 questions quickly about equity, and that's something I know you all care about quite a lot and have done some work on. So how do we promote universal access to analytical tools, data analytical tools, including in low and middle income countries?
I believe, Shyam, that the development of open source tools and user-friendly is fundamental for the democratization of data analysis. And the availability of public web servers is also crucial, since many of the teams, they do not have access to the computational resource to run their own analysis. And also build capacity in underrepresented region is also extremely necessary. We have we had a situation, at least here in Chile, in Latin America in general, that most of the scientific community, they did not know what single cell means. This is the first key point. There is a lot of people. There is still a lot of people who don't know what single cell analysis, single cell data means. This is something very key.
Absolutely. You brought up the issue essentially of training and, okay, Neil, let me go to you first. Did you?
Yeah, sure. Yeah, I just want to add, I think that's a great point. I just want to add 2 points to this. So the first one is really, we already touched upon this point of reproducibility, right? So I think that's a big part of it, right? So making available the data in a way that is easy to acquire, with fully reproducible scripts and so on that are well documented with all the lingo and everything. I think that's an important part and a part of the responsibility of people who publish in this in this domain. So for example, you can think about just making available the count matrix in its final form with the latent space and everything. So you don't need to run again through this whole processing process that takes a long time and compute power. And one more thing to add to that is also in building tools, right? So it's a good, I think it's going to be a moving forward good habit to think about compatibility with cloud platforms that are either free or not very expensive, like Google Colab and others.
I want to raise one point actually, because this is not just the issue about LMICs and people without access. I mean, in many ways you could see it, within the communities that we're actually physically interacting with. Maybe from the perspective of the clinical communities, where, the more you want some form of ability to sort of like navigate through the data and make some queries, but without necessarily the kind of heavy bioinformatics, that's required, which is the case at the moment. So how do we get or shift it so that it goes further along the ability to browse, ability to query, but you don't necessarily have to be a computational biologist to do it.
You're bringing up the issue of user-friendliness, essentially. And absolutely, the atlas will only be popular and widely used if it's user-friendly. And if it can be used by people who are not professional bioinformaticians. So, excellent point. And Vinicius, you brought up the issue of training. You want to just quickly touch upon your experiences from the wonderful workshop you co-organized, HCA Latin America? Yes,
it was a really great experience. This was a workshop focused on RNA-seq data analysis. It was a 2-week virtual workshop in which along the first week, we had introductory talks for a broader audience with about 400 attendees. And for the second week, we selected 30 people from different countries to work separately with teams from Brazil, Chile, and Mexico in a hands-on, very intensive course along the whole second week. The results were amazing. Novel collaborations started. For example, different groups worked together in proposal for the last CZI Ancestry grant. And even a poster was submitted to this event here and was presented today. They developed the poster, all the data analysis along the workshop. it was a particular group that participated, and with the data they analyzed along the course, they submitted to the event. And I believe that this kind of this current situation with all the virtual world is a great opportunity to this kind of training, because we have the opportunity to have great speakers participating along both weekends from abroad, from the whole world. And I believe this could be a reference to other courses for underrepresented regions. And another key point related to training, I believe is something that the International Society for Computational Biology do a lot, is to provide, to raise discussions on providing standardized data sets and guidelines to be used specific for training. This is something that would be very helpful. For us was very complicated at the beginning, a lot of work to define everything, but I believe it was perfect, worked perfectly.
Absolutely, I attended it, and I really want to congratulate you guys for pulling off really a landmark event. And I think many more such workshops need to be held to popularize, democratize single cell analysis and the work in the Human Cell Atlas. So it was quite a landmark. And thanks to Christine and everyone else in the HCA team also for a very professional organization of that event. We have some very interesting questions coming from the audience as well. So I'll give you all just one sentence each to address the last question on our menu before we move to audience questions. So just in one sentence, spatial omics, what's the most important computational challenge? Muz?
It kind of depends on which spatial omics, because they...
Pick one.
I guess if you were to take the one where it's not quite single cell, but has the most number of transcriptomes, something like spatial transcriptomics, how do you kind of like really deconvolute it to be able to work out what is the single cell level expression? That's a challenge. There are algorithms out there, and things are moving very, very fast. But as always, whenever there's something new, there are just so many options and you don't know which one to go for. And by the time you learn one thing, you realize that you've kind of like learned something that's now superseded by something better, and you have to relearn it again. So lots of challenges for at many levels.
Couldn't agree more. Chris?
Without a doubt, I think it is that we have to be cautious about not over-interpreting the beautiful patterns that we're seeing. I think there is a lot of really spurious correlation coming out of the area, although a lot of really fantastic innovation as well.
Avoiding artifactual patterns. Neil?
Yeah, so I guess I'll mention two things very briefly. The first one is to use that spatial data to contextualize what we see in the standard single cell RNA sequencing and see really how we can map what does it mean and what types of effects do we see from the microenvironment of the cell, and then it gives us a better tool to look back at our single cell data and interpret it. And the second thing is something that is, I guess, would become a necessity as this field evolves, is to try to start doing comparative analysis or differential analysis, if you will, between samples, but looking at phenotypes that are more abstract than gene expression.
Differential spatial analysis, that's certainly a frontier. Vinicius?
Yeah, it's quite similar to all the answers. It's important to have improvements in the methods modeling the relation between space and expression data in order to define changes that can be quantified in a tissue. For instance, the spatial identity in the neighborhood of particular cell types, for example, across tissues, conditions, and diversity.
Got it. Awesome. All right. Lots of great questions from the audience as well. So I'll just read out the question, and then whoever wants to take it, please raise your hand. This is from Bruce Aronall. Terrific discussion. Temporal seems to be less well modeled in HCA schema than one would desire. How to improve that? So how to improve temporal modeling? And also per cell, how can a standard be developed for its position in a trajectory? Tough question.
Yeah, so maybe I can chime into that. Two things. The first one is, of course, the temporal view should be used when it's relevant, right? But I guess it is in a subset of the HCA projects, right? So, and you know, there is a lot of activity, a lot of development in this area. I know a lot of people are working on it, and we've heard some of these talks in previous HCA meetings. So I think there's definitely some action going on there. But it is very difficult. And it's also very difficult to validate the predictions that come out of it. So it's definitely a challenge. There are some technologies that are opening up now that make it you know, one step further. I don't know about implementation of those in the HCA, but now there is this single cell level lineage tracing with CRISPR-Cas9, which I think is very promising area. But of course, just to incorporate it in you know, human cells, so that's going to be very. I mean, of course, it has to be in vitro, so that's a completely different set of challenges.
Thanks, Neil. Can we say that the temporal modeling that or representation of temporal trajectories that Bruce brought up, can we say it's in a sense a subset of the broader problem of representing any continuum of diversity in the human cell atlas? Instead of a temporal continuum, it could be in, you know, in, let's say, over hours in one sample. It could be a continuum of age. It could be a continuum of ancestry. It could be any continuum that we want to represent, right? And I think they're all the same problem in terms of representation, in terms of modeling. And it is, I would say, a very, very important challenge for us in the community.
I want to add one point about this the issue of temporal. I mean, often what we do is actually take snapshots. It's not, we don't, in the kind of like human cell atlas referencing or profiling, you don't actually necessarily have data of one individual across time. What you're doing is actually taking different snapshots and then stitching it together. So it actually has a second complication to actually inferring temporality within these type of data sets that may need to be taken into account of.
Absolutely. Yeah, it's like phylogenetics or looking at the fossil record or something. You have one landmark, one fossil here, one fossil here, one fossil here, and you somehow construct a continuum through that. So we are doing interpolation and stitching things together because they're near each other very often, if you don't have actual lineage tracing data. Jump quickly to the next, one more question from Asaf Rotham. How do you address missing metadata? Is there any system to feedback on the quality of sample and metadata sets? So what is the quality of the, say, cell by gene matrix? What is the quality of the metadata? How do we, I guess, quantify that, automate it?
It would be super useful to be able to comment.
Sorry, Chris, I interrupted you.
It would be super useful to be able to vote up or vote down data sets on their quality. I don't know if that's possible within the data portal.
Right. Okay. Yeah, so some kind of community rating. That makes sense. And I'm sure, yeah, all our social media people are very familiar with that.
Or I guess in terms of publications, because the data sets that usually have good quality data and metadata are often reused, and you can actually see how often other people have used that data set. I mean, that's also an indicator, I suppose, as a feedback.
Absolutely. Usage is the biggest endorsement. Any other?
I do think, Shyam, that we'll get to a point where we can infer some metadata just because we'll have sufficient depth of data that will be able to build correlates that are missing. But for the time being, I think constructing the atlas is going to be really constrained by a lack of good quality metadata.
And also, I believe this is something that we need to motivate data repositories and journals to consider. This should be a requirement for prior submission. Definitely, in my opinion.
Which comes down to the question of minimal metadata standards. I think we just have to get there to have a usable atlas. All right, on that note of all being responsible citizens of the single cell world or the human cell atlas world and submitting all our data in good shape, I think that's a good note to end, and we're bang on time. So thank you very much, all of you, Moz, Vinicius, Chris, Nir, and everyone who submitted questions, and all the other people whom we can't see here, but were watching this panel. Thanks a lot.
Thank you, Shyam. Thanks, everybody.